The overall mechanism of how DNA replication is carried out will be similar with respect to prokaryotic and eukaryotic cells, meaning that you will have an origin of replication, one or multiple. This will form a replication bubble. You will have a replication fork, and much of the machinery that is involved, or enzymes and proteins involved in this process, are highly conserved and similar between prokaryotic and eukaryotic cells. One of those enzymes is DNA polymerase. This is the important enzyme that is responsible for creating the new DNA strand. It does so by adding the complementary nucleotides to the new DNA strand using the parent DNA as a template. DNA polymerase needs a DNA template in order to perform. It also needs a primer in order to know where on the DNA to attach to. This is usually inserted at the free 3' OH end of the growing strand. Please note that DNA can only read and create a new strand in the same direction, meaning that it can only read the template strand in 3 to 5. That means that it can only make a new strand in the 5 to 3' direction. You will commonly see that DNA polymerase will work in 5 to 3. However, I find that it is important for students to understand and break down this process into two steps: the direction that DNA polymerase can read in, and the direction that it will make the new strand. The nucleotides are then added to the growing DNA strand. However, these nucleotides usually start out as in triphosphate form, meaning that they each have 3 phosphate groups attached to them. During the process of DNA replication, DNA polymerase will be able to cleave off 2 of these phosphate groups and be able to insert these nucleotides in monophosphate form. Note that in the process of DNA replication, we now have DATP, DCTP, DGTP, and DDTTP. These refer to the fact that deoxyribose sugars create the base in these nucleotides. These are very similar to ATP, however, remember ATP has a ribose-based sugar. Once again, ATP is not the only energy source for the cell. DNA polymerase can extract the energy from these phosphate groups, and this can be used to drive the process of DNA replication. This slide depicts the action of DNA polymerase. Note that we have a template strand that is running from 3 to 5. Also note that the formation of the new strand will thus be in the 5 to 3 direction. Note that the incoming nucleotide has 3 phosphate groups attached to it. DNA polymerase can catalyze the removal of those 2 phosphate groups, creating a monophosphate nucleotide. At this point, it can now be incorporated into the growing strand. The replication fork refers to the bubble that forms at the origin of replication. 2 replication forks are present for each replication bubble. Replication will proceed at both forks in opposite directions. Remember the requirements of DNA polymerase. It needs a DNA template, and it only works in one direction, meaning that DNA polymerase can only ever read in 3 to 5, and it will only make a strand in 5 to 3. This creates a problem because while at each replication fork, one of the strands will be presented to DNA polymerase in the right direction. However, the other one will be in the opposite direction. Also remember that a primer needs to be inserted in order to have DNA polymerase function. This primer is commonly added by DNA primase. Note that this primer is also considered to be an RNA fragment, meaning that the base sugar in that molecule would be ribose, as opposed to deoxyribose that we see in the DNA molecule. Each strand can be referred to as the leading or lagging strand. The leading strand is one that is present in the right direction. As a result, when DNA polymerase binds, it can continuously copy that strand as the replication fork increases and the DNA becomes exposed. However, the other strand we refer to as a lagging strand. This is a strand that is running in the wrong direction. As a result, it will be copied in small fragments called Okazaki fragments. These fragments will eventually be sealed together using an enzyme called DNA ligase. We can see that there are many enzymes and proteins involved in this complex process.